Hi everyone, Brenda Gustin here, and this is our first lecture video that you're going to be using to complete your lecture study guide for week one. Remember that you can use this lecture video or you can use your ebook or a combination of the two to complete those lecture study guides each week. So we're in chapter one. All the information from this week is taken from chapter one, and you'll notice on your lecture study guide that I have section 1.1 listed. So I underline the learning objectives or learning outcomes. So you can see right here, here's one of the learning outcomes under section 1.1 on your lecture study guide. The first thing it says is describe the levels of organization of life. And so the first thing I would do if I were you is I'd start memorizing from the simplest to the most complex. How do we get to a living organism? And we start with atoms. We go to molecules, cells, tissues, organs, organ systems, and then organism. All of those are listed in your under the vocabulary and in actually the learning outcome in section 1.1. But let's make sure we have an understanding of what each one of these mean. So all living organisms are the most fundamental parts that they're made of are atoms, and we're going to talk about chemistry in the next module. But remember, the atoms are made up of the unique elements. And so in this molecule of water, we have three different atoms. We have one molecule of oxygen and two molecules of hydrogen. So two different elements, or three units, three atoms. And you might remember from some chemistry somewhere that when atoms combine with each other, they do that through chemical bonds, and they form molecules. So these three atoms came together through chemical bonds and formed a molecule. So all living things are composed of atoms that connect to each other to form molecules. And then these molecules come together to make cells. And the cells are the smallest, it's the smallest structural and functional unit of an organism, meaning it's the smallest thing that can be alive. A cell is going to be your smallest possible living unit. So there are some organisms that are composed of just one cell and others that are multicellular, like us, made of lots of cells. So when we take cells, and so here's a cartoon of some I think this is actually supposed to be skin. So here, each one of these units here is a cell and making up the skin. And you're probably familiar with the nucleus and all that. So lots of cells here. And when we have lots of cells that come together, groups of similar cells performing a common function, we call these tissues. And again, we're going to have a future lecture where we talk about cells and we talk about tissues. So right here, these are lots of cells performing a common function. And because this is skin and it covers a surface, it happens to be epithelial tissue. And we'll talk about that this semester. Continuing on, groups of tissues, at least a couple tissue types come together and make organs. So hopefully you recognize this organ here, that's the heart. And the tissues that are present in the heart, there's mainly muscle tissue, but there's also some connective tissue that holds things together. There's epithelial tissue that's on the surface, and there's nervous tissue that's controlling the electrical impulses. So organs are composed of several tissue types. They have distinct shapes and distinct functions. We're going to talk about a lot of organs this semester. Multiple organs that come together form organ systems. We kind of organize them for understanding into organ systems, or groups of organs that work together for a common purpose. So if you think about what organ system is the heart in, that's in the cardiovascular system. So here's a diagram of the cardiovascular system. There's the heart. We've got arteries and veins and making up the cardiovascular system. And we'll talk about 11 different organ systems this semester. And finally, the organ systems come together to form the organism. And in this course, the organism is the human body. There's a picture, figure 1.2, that goes into that same detail that I just did up to here, and then goes even beyond that into the ecological level of how organisms fit in with the environment. But for this course right now, we're going to be sticking with from atoms to humans. So you should be able to state each one of those steps and what each of those mean. Okay, our next learning outcome, if you look at your lecture study guide, is in section 1.3. It says describe the general purpose of the scientific method. The scientific method is a standard series of logical steps used to study and understand the natural world. You use the scientific method in your daily life, whether you realize it or not. You might use it, some might use it more effectively than others, but when we're looking at the scientific method, it's the way we learn about the natural world. And the first thing you're going to do is you're going to make an observation. We're going to create a hypothesis about that, and I'm going to go over each one of these steps in the future slides. That hypothesis is going to be tested in an experiment. The experiment is going to yield results in the form of data. So we're going to get results of that experiment, and we're going to be able to draw some conclusions. So we talk a little bit more about this in lab this week. And one of the things I emphasize in lab is that the scientific method kind of keeps cycling back on itself, because a lot of times what happens is we make conclusions, and that causes us to have to make a new hypothesis and go through this process again to really keep testing different aspects of our observation. So I'm going to kind of expand on this. So here's our observation. An observation is a formal way of watching the natural world. So it happens all the time. We're always using our senses, our sight and smell. In research labs, we might use some instruments, all sorts of instruments to observe things. And through research and data, we acquire new observations. The thing about our observations is that these observations should be objective and not subjective. And what that means is I think of an object as being something solid that I can touch. And so I think of objective observations as being factual. And what that means, objective versus as opposed to subjective, is I can make an observation and think someone is very beautiful. But it's very hard for me to test that because beauty is subjective. And beauty is going to be somebody's opinion and somebody's judgment, and different people have different ideas of beauty. I can't really test if someone is beautiful with the scientific method. What I could test is the characteristics of somebody that is perceived as beautiful. So I can, ask 100 people which person is more beautiful. And then I can analyze the characteristics of that person and try to determine the characteristics of beauty, which would be objective, would be factual. What do people think is beautiful? I can test that, but I can't really do an experiment and test, which person or which people are more beautiful, because it involves judgment. So once we made an observation, we're going to develop a hypothesis. And a hypothesis is going to be a possible explanation for the observation of a natural event. It must be testable. So we have our observation. Let's say my observation is in the winter my skin's really dry and it's real flaky. And so my hypothesis might be that drinking 64 ounces of water a day will prevent my skin from forming dry flakes. I might be thinking, maybe I'm dehydrated. So what if I drink enough water each day? Will that prevent my dry flakes from forming? So my hypothesis has to be an explanation for my observation, and it must be testable. Drinking 64 ounces of water a day will prevent my skin from forming dry flakes. Or another hypothesis for a different problem is sleeping 8 hours, for different observations, sleeping 8 hours a night will make workers more productive. So there you go. Various hypotheses, things that can be tested and can explain my observations. The next learning outcome I have right here, explain the importance of control and experimental groups and variables to an experiment. So we talked about an observation, and then we went to a hypothesis. So now in the scientific method, the next thing we're going to do is we're going to create an experiment. And to create that experiment, we need variables. And variables are going to be things, and there's different kinds of variables. Here it's telling you there's experimental variables and responding variables. But variables are going to be the things that change in the experiment. So for example, if I'm going to be doing the experiment on, if I'm going to test if drinking water causes fewer flakes on my skin, the experimental variable, the thing that I can modify, it says the variables are modified by the researcher. Experimental variables are modified by the researcher. I'm going to get a bunch of people and have them drink different amounts of water. The responding variables are going to be the results. So now we're going to see after a period of time, if the people that are drinking different amounts of water have different amounts of dry skin, amounts of flaky skin. The other thing you need in an experiment are a test group and a control group. So the test group is the group where we're giving them different amounts of water and we're testing their flaky skin. The control group gives us something for comparison. Let's say drinking the different amounts of water did have an effect on the skin. But we don't know if it really could have been the amount of humidity that the people were living in. So maybe we need a control group of people. We need a control group of individuals that remain at remain at constant humidity, because we can't keep people at the same humidity all the time. So we might need a control group that doesn't change their amount of water, but changes their amount of humidity. So that could be something to compare to. So the control group are the test subjects who are not subjected to the experimental variables. They don't get a different amount of water. They're all drinking the same amount of water, but they're being controlled for other environmental factors. So a control group is needed for comparison. So let's see, observation, hypothesis, experiment, and now we have our results. And our results are derived from our experiment, and they come in the form of data. Data usually is numbers. We try to, compare numbers to each other. It's because we can analyze numbers statistically, but in some cases your data might be things other than numbers. It might be photographs and descriptions and qualitative things instead of quantitative things. But somehow we collect some data from our experiment. Related to data, another objective is recognize the importance of statistical analysis to the study of science. So statistics are mathematical formulas used to interpret data, and they're a way to analyze our data to see how accurate it is, or not accurate, but how certain it is, how likely it is. Statistical data include a standard error, also standard deviation, to show how uncertain a value is. It says statistical significance is determined to evaluate the probability that the results are due to chance or some other experimental variable. So, we know in biology, there's a lot of variation. Day to day, you feel different. And so we want to know are the variables in our experiment actually causing the changes in the experiment? And the only way we can really figure that out is through using statistics. Here's an example. This is in your book, figure 9.1, and it's showing you here they're comparing blood cholesterol over time. And they must have done something to, they're just monitoring blood cholesterol. And so here we go, that they're showing you, here it is over time, and you can see it went up and then it went down. But they've done statistics on this to show, well, there's actually a range, because usually you don't do experiments on just one person. You do on many people. And so they find this range over time for these individuals. And these statistics that you're doing, here we go, the standard error also causes standard deviation, is going to evaluate, is this difference resulting from chance or from other experimental variables? So statistics are really important in determining kind of the credibility or the believability of your data. How compelling is this data? Is it convincing? Then our last step in the scientific method is going to be to develop a conclusion. So we're going to analyze the data, and then we're going to draw a conclusion. So from here, you might conclude that over this period of time that the cholesterol on average went down. And so our conclusion is going to use our data, and we're also going to talk in our conclusion about whether our hypothesis is supported or not. And so how true is our hypothesis, or how much do we have data that supports our hypothesis? Not really how true it is, but do we have data that supports our hypothesis? And a lot of times what happens is when we make a conclusion about the experiment, then we're going to develop another hypothesis and test some other aspect. So the scientific method kind of goes in a cyclical way. The more we learn, the more we need to test, and the more details we learn about a problem. Here's another learning outcome in section 1.3, and it's recognize the importance of scientific journals in the reporting of scientific information. So once we've done an experiment, in fact, usually you do many experiments, and you test a hypothesis and you come up with some compelling conclusions, you want what happens to them? Well, in a lot of scientific studies, the results and conclusions will be published in scientific journals. And you can see here's one called Nature and one called Science. These are two of the, if not the two most prominent science journals in the world. If you get in Nature or Science, you've done something pretty important. But there's lots of other journals: Journal of Bacteriology, Journal of Cardiac Care, Journal of there's a journal of everything: Journal of Immunology, Journal of Asthma and Allergies. There's like journals of everything. And to get your work in a journal, these are usually very scientific, and they're reviewed by experts in your field before they're published. This is going to be some of the most reliable scientific information you're going to be able to find in journals. Most of us are not really at the understanding level of science to be able to read these and interpret them. And so there's lots of other sources for us to find out scientific information. And that can be from magazines, like things like science magazines like Scientific American or Discover or National Geographic. You might find a lot of your scientific information from websites. And so one of the things I just want you to be aware of is the information that shows up in different documents can have different quality. You can probably assume that the information showing up in scientific journals like Nature and Science is of the highest quality. It doesn't mean there's not mistakes made, but it's of the highest quality that we can test for at the time. If you're looking for things on web pages, you've got to be careful at how you're going to evaluate it. And one way to really look at it is to look at the last few letters. Is it .gov, a government agency? Is it .com, a commercial agency? And that could really depend. Is it .net or something like that? You've got to be careful of where you're getting your information to assess the likelihood that it isn't biased or inaccurate in some way. And a lot of commercial things do have reasons that they're selling things, so they kind of lean their scientific information in one direction or another. So you have to be aware. The last objective I have here is explain the importance of a scientific theory and give examples. I think a lot of us think of the concept of a theory and say, oh, that's not a big deal, it's only a theory. The word theory that we use day to day is, I have a theory that when I drink too much coffee that my stomach gets kind of unsettled. I have a theory that it's how concentrated the coffee is or something, I don't know. And so we kind of take that as light, there's not a lot of proof for it, I'm just kind of thinking about it. But in science, a theory is the best we have. A theory is a widely tested and widely accepted explanation for something and how it works. So a theory is a really big deal. I've actually heard people say, oh, evolution, well, that's only a theory. But if you're a scientist, only a theory means it's been tested over and over and over again. And there's lots of different angles on it and lots of data. And it's really as close to getting absolute proof. Now, how would we get absolute proof for evolution? I don't know. Maybe you have to be there when it started. So the best thing we can do is a theory. Here's some of our common theories. A cell theory, that all organisms are made of cells. So far, it's held true. Theory of evolution. Life changes over time as a result of natural selection. Germ theory of disease. Microorganisms can cause disease. Like all of these different things are theories, and they're very critical to the understanding of science. I'm going to stop right there, and I'm going to finish this lecture in the second video.